Okay, we resume with our introduction to the limbic system. What do you know so far about the limbic system? It is part of the triune brain, a conceptualization of three layers in the brain, and it's functioning, and don't take it too seriously or literally, but basically referring to the most ancient-like structures down at the base of our brain that we share with every other vertebrate out there, especially those reptiles, and just automatic endogenous functioning of things, keeping track of your blood pressure and your blood glucose and body temperature and things of that sort. Then the second layer, the limbic system, and then on top of it, the magnificent, gleaming elitist cortex. And the key thing that should have come out of the lecture the other day was, amid the temptation to go with this model of top-down regulation, the cortex tells your limbic system what to do. And that very frequently takes the form of your cortex trying to convince your limbic system not to do something disinhibited and impulsive because you're going to feel sorry for having done that eventually, and your limbic system in turn sending messages down to your reptilian brain. Oh my God, you can have an emotion and your heart beats differently. Go try that. If you're a Komodo dragon, that's the limbic connection there. That's great. But as we saw, just as importantly is bottom-up regulation. Your body temperature, your breathing rate affecting your emotional assessments of things, how hungry you are affecting decisions you make, and probably most importantly, the ability of the limbic system to talk to your cortex just as readily as your cortex talks to your limbic system. Downside of that, that's the circumstances where, under extreme emotional duress, you make some horrendous decision and do something impulsive, and it's a disaster, and you have the limbic emotional brain driving your decision-making cortex. So some of the time that's a disaster. What we will see later in the course is some of the time that is great, because that's the means by which you get the cortex to make decisions that are empathic, that are feeling somebody else's pain. And we'll see when the limbic system, because of damage, loses the ability to talk to the cortex, you get people who function like machines and make horrifically cold, heartless, utilitarian decisions. Okay, so we've got crosstalk between all of them. And what we were left with was what every single structure in the brain wants to do, which is to be able to dominate everything else going on. And within the limbic system, the two main desires are for all of these limbic structures to tell the hypothalamus what to do. Why is that interesting? Hypothalamus, which is sort of at the transition from limbic system to reptilian brain. Let's just give us a spinal cord here. Okay, so there it is. And why does everybody want to control the hypothalamus? Because it controls everything else going in your body by way of hormones coming out of your pituitary and by way of your autonomic nervous system going down to your spine. So one desire of limbic structures is to regulate stuff going on throughout your body by way of hormones and autonomic stuff. The other great desire of the limbic system is to tell the cortex what to do. And most of all, the frontal cortex. Those are the goals, those are the secret aspirations of every limbic structure, to control the hypothalamus, to control what the frontal cortex is doing. And now what we must do is, in every generation, is the duty of a neuroscientist to inflict limbic circuitry on the uninitiated. And this is like magnificently obscure and multisyllabic, and everyone needs to go through this, and not just to get your neuroanatomy secret handshake information, but to get a feeling for how you wind up getting conflicting emotions and mixed emotions and ambivalencies and all of that, having much to do with all of these areas of the limbic system jockeying each other for control, for influence. All right, so we start off with this, and as was termed around the time people were figuring out the nose brain, and is the same thing as the emotional part of the brain, and deciding to call it the limbic system. Limbus, Latin or Greek or something for edge, limb, I don't know, something or other. All of them get names like this. But into the limbic system in the 1930s leapt this obscure neuroanatomist named James Papez. James Papez, P-A-P-E-Z. And nobody knows, including me, how it's pronounced, whether it's Papes or Papez. And what he showed was there was a whole interconnected circuit of all the limbic structures, now known as the Papez/Papez circuit. And he did that and was never heard from again. And this is what we have. This is the different structures within the limbic system. Structures. From the other lecture, you got a whole bunch of neuron cell bodies all clustered around that are functionally related and are sending cables of axons, projections to other structures, nuclei. This is what we'll be talking about, the gray matter specific structures in the limbic system, and then the projections, the cables, the axonal whatevers, the white matter stuff connecting different structures with each other. So we start off with what we already know, the hypothalamus sitting down here, which is sort of honorary limbic system if you're a reptile, or if you're a mammal, you decide that is only honorary part of the reptilian brain, but it really belongs to us in the limbic system. Hypothalamus as this portal, incredibly important, and hypothalamus not a single nucleus, but instead made of 20 different subnuclei, and you come back from a long weekend and there's 3 new hypothalamic nuclei that have been identified, and we will see they are really important for a lot of the stuff to come. Other structures, the amygdala, which will figure in our lives from here on out, the amygdala sitting here, the hippocampus, which is the greatest part of the limbic system, and this could be proven scientifically, the septum, something called the bed nucleus of the striae terminalis. And I hope people are not scrambling right now to write all of this down. We will, in the extended notes, give you diagrams that will save you from this. Coming around to something called the mammillary bodies, ventral tegmentum, nucleus accumbens, thalamus, and then sitting on top of it, the whole cortex, with our favorite part from now on being the frontal cortex. And the person's nose is on this side, they're looking in this direction. And thus, shockingly, the frontal cortex is at the frontal part of your brain. Okay, so we've got all these structures here with their agonizing long names and stuff. Amygdala, I was under the misconception for many years that that was Latin or Greek or Pig Latin or something for peach pit, until someone informed me, no, that's actually the word for almond. And when you look at it, it kind of looks like an almond. Hippocampus looks like a seahorse. A hippocampus looks like a seahorse when you look at it, and everyone decided thus to call it that. River hippo, Mesopotamia, I'm free associating here. Hippocampus, seahorse, and yeah, it kind of looks a little bit like that. When you cut it in cross-section, when you cut it like this, it actually looks much more like a jelly roll or a cinnamon bun. And that is always on your mind when you look at the hippocampus. Hippocampus, septum, I don't know, divider, thalamus, which shockingly is above the hypothalamus. And shockingly, the hypothalamus is underneath the thalamus, these other areas. Okay, the first thing to begin to process is a lot of these structures, they are coming in twos, in pairs, bilateral. Your brain is bilateral. You got your hemisphere here, you got your hemisphere there. We saw that bizarre thing the other day, that motor projections from this side of the brain come down and innervate that side of the body, crossing all of that, but two hemispheres, and thus some of the structures you get two of. There are two amygdalae. Two amygdalae, and if you cut through the brain here in cross-section and look at it, looking at it here, here are the two amygdalae there on either side. And we will eventually see the amygdala has an inner core that has a very different function than the outer core. This will become very relevant to who we decide we hate and who we instinctually are afraid of and things of that sort, and how you could learn to stop having those feelings about someone who counts as a them right now. We will get to that. Okay, two amygdalas, two hippocampuses, two hippocampi, and now we're cutting in cross-section here, and there we are. There's the two hippocampi. Notice we now go to the septum for the moment. There's only a single septum. Septum, center. The divider in your nose is your nasal septum. Your heart has a septum dividing the chambers on this side from that side. The septum is this brain nucleus sitting right in the center there. And what we see is somehow the hippocampus is going to get around to the septum. Two hippocampi to one septum. What's that about? Two hippocampi, let's move further back, begin to merge towards each other and then eventually dive down into the septum as one. So two hippocampi okay, which direction here? Coming up and merging together into one and dropping into the septum. The other name for the hippocampus is Ammon's horn, some mythological whatever, but like a ram's horn, that sort of thing. So there's one septum, there's one thalamus. We're now cutting through here, one hypothalamus. We're now in this section, and we have accumbens, thalamus, the two hippocampi, hypothalamus, and pituitary. And then heading further back, the cut through here, we've got two mammillary bodies, one ventral tegmentum, and here we see the hippocampus that is coursing up this way and should have been labeled. So hippocampus merging down in there. All right, so just to orient you in those ways, what we now begin to look at is the dread papes circuit, where what you're going to see is essentially every one of these structures talks to every other structure, and where all of them are trying to find shortcuts to tell the frontal cortex and or the hypothalamus what to do. Here begin the agonizing details, and these will be relevant for the rest of the course. Number one projection, the amygdalofugal pathway. Amygdalofugal, what it is most importantly doing is connecting the amygdala and the hippocampus. And we will see there's some very striking circumstances where the amygdala gets emotionally worked up enough that it takes over the hippocampus. And we will see what that has to do is traumatic memories, highly traumatic, that will stay with you forever after. And because the amygdala is kind of this unmodulated hothead, the memories you form about that trauma may not be terribly accurate. We'll be getting to that. Okay, so amygdalofugal pathway. What's number two on our list? The striae terminalis. Okay, here we see the whole lesson from the other day about bizarre stuff that could happen in terms of embryology. So the amygdala talks to the hippocampus, which, as we'll shortly see, talks to the septum. So the amygdala wants to influence the septum. Here it's one synapse, two synapses away. Maybe the hippocampus has some thoughts about the matter and will distort what the amygdala is saying. What the amygdala wants more than anything, however, is to tell the septum directly what to do. And number two, what is called the striae terminalis, bidirectional. Watch this, goes like this into the septum. Oh my God, could you have a less efficient way of wiring things up? This is back to our theme embryologically, where at one point the septum is down there and something folds and it's up here, and you got, so this striae terminalis. Notice the most relevant functional implication of this is the amygdala is 2 synapses away from the septum. By way of the amygdalofugal pathway, it's 1 synapse away by way of the striae terminalis, and that's what it takes advantage of very frequently to override the hippocampus. All of this ending in, more specifically, the dead nucleus of the striae terminalis. This is where these influences wind up. Okay, so at that point, we've got an amygdala and hippocampus, either in tandem or in parallel, talking to the septum, bed nucleus, striae terminalis. We are going to see there's all sorts of interesting things going on with this, having to do with sexual dimorphism in the brain, and we will see having to do with sexual orientation, sexual identity. This will become an interesting region. Okay, so at this point, you've got information flowing down. Our next thing, the medial forebrain bundle. The medial forebrain bundle is this big honker of a projection that goes to the hypothalamus and goes through it and then continues on to the mammillary bodies and then on to brainstem areas. Medial forebrain bundle. And what we've now got is flow of information. And notice, critically, all of these projections are bidirectional. Everybody gets to talk to everybody else. And in this case, this is now funneling the amygdala and hippocampal information by way of the septum and the bed nucleus striae terminalis, heading it down now to the hypothalamus, where its desires lurk to finally control everything going on here. Good. Next one, we've got the mammillothalamic tract. Let's really cut loose and change colors here. The mammillothalamic tract goes from the mammillary bodies to the thalamus and goes here. And what we're going to see is nobody really knows what this does, and everybody knows what the thalamus does, and it is so boring in lots of ways that I don't even know why it winds up in the limbic system. We are almost going to entirely ignore the thalamus for the rest of this course, except for one really critical thing that it does, in that occasionally it could send information about a threatening emergency to the amygdala in a fraction of a second before your cortex even knows what it is that's going on. It could do it in implicit, subliminal, incredible speech. We will eventually get around to that. And that has a whole lot to do with the world in which you get somebody in a uniform who winds up shooting some unarmed person and is convinced that the cell phone the person was holding was actually a handgun. And we will see that has a whole lot to do with this shortcut we will see by which the thalamus talks to the amygdala. From there, we've got projections from the ventral tegmentum. Okay, we need 2 colors here, so number 5. The ventral tegmentum sends a bunch of projections to the limbic system. The mesocortical projections here, it goes here, and it goes there, and it goes there, and it goes everywhere all over the place. And we will see that's mighty important, because what the ventral tegmentum is about is reward and pleasure, and we will see where that fits into all sorts of stuff. But then, in addition, the ventral tegmentum has the mesocortical projection, which, as you would guess now, goes here and is going to be super relevant. So mesocortical, mesolimbic, Okay, so ideally you're completely overwhelmed by now. Again, you'll get diagrams of this in the extended notes. What should you take from this? The flow of information down to the hypothalamus, the flow of information getting to the frontal cortex this way. And just to horrify you, there's a whole bunch of other structures in here that are relevant. The basal ganglia, the habenula, things like that, that I'm not torturing you with. And everything connects to everything else. These are just the most dramatic projections in there. So every single one of these areas has the means to directly talk to each other. Talk to each other, sending axonal projections that are synapsing with their axon terminals onto this next region there. All right, so we've got this whole agonizing diagram here. Just to go through again, cup through number one here, and you get two amygdalas, you got your one septum, and you just got the tail end of the frontal cortex, cup through here. We have right in the middle of the limbic system, cut through at the back end, you see the arch, the horn of the hippocampus at the back end of it, and is soon going to come down and dive down into the septum. And it will do so, one last projection here, it will do so from the hippocampus into there by way of something called the fornix. And the fornix just shows you how fun neuroanatomy can be. Fornix, arc. It's the arc that picks up the horn here, this almond horn, some sheep in mythology, pick up on it and turn into this arch that goes down to the septum. Fornix, Latin for arch, and great bit of etymological history. Fornix, it used to be in ancient Roman times. In Rome, prostitutes would hang out at the Colosseum under the arches, and thus the term evolved that you were going off to go do some arching, you were going off to fornicate. Yay, isn't neuroanatomy irresistible hijinks? So we've got all these connections now, and these are going to be highly relevant to all sorts of stuff down the line when it comes to what happens when this stops working or this gets overstimulated or things of that sort. But we transition to now is what this is all about functionally. So first off, how do you find out what the functions are of the limbic system? And this is really, really tough because what you would eventually see in med school is you can localize, here are the motor neurons that are responsible for moving this finger. And here's the sensory motor cortex that's responsible for if you're having tactile stimulation. There are things that's incredibly localized, so you could see somebody after a stroke by what parts of their body they can move, they can't move. You can exactly localize where the damage occurs. That's where do you exactly localize nostalgia? Where do you localize jealousy, where you know you shouldn't be jealous and you should be more mature than that, but nonetheless you were jealous? Where do you localize poignancy? By the time you're getting to the limbic system, trying to figure out what these parts of the brain do are so much harder than these are the three neurons back here that tells you you're looking at a dot in this side of your visual field. It is incredibly messy when we are dealing with our emotional brain, our social brain. So what are the techniques? Here's one of our limbic structures made up of a whole bunch of individual neurons forming a nucleus accumbens back on the other side, for example. The first approach, the classic approach, the easiest one is to go in and lesion it, destroy this part of the brain, and surgically, chemically, there's all sorts of different ways of doing that. Destroy that part of the brain and then see what doesn't work right anymore. And you've just now learned something about what this part of the brain does. And the lesion approach is what taught people about neurological function for centuries because of the happy circumstance of how many damn wars we've had where people wind up getting missile wounds. That's the clinical term for it. Something bashes and destroys part of their brain. And now you see what doesn't work right anymore. And that led people for the first time to begin to think, oh, when you blow out this part of the brain with like your bow and arrow or your shrapnel or whatever, this doesn't work right anymore. Whereas when it happens here, that doesn't work. There's localization function. So the crudest way of finding out about this is just go in and destroy this part of the brain. And what happens? What happens? And that's one way of learning what's going on here. Next strategy, you could do exactly the opposite. You've got this part of the brain that's sitting here quietly. It has absolutely nothing to say. It hasn't had a thought, whatever. And you stick in an electrode and you could stimulate this part of the brain. Give it the false belief that it is being stimulated by neurons elsewhere. You stimulate that part of the brain and see what happens. What happens at that point? And it is very clear. You go and stimulate this tiny part of the brain, and you now do this with your finger. That's pretty clear news as to what's happening. Here, stimulate this emotional part of the brain. You're going to have to be really good now at figuring out what's working differently. So, lesion strategy, stimulation strategy. Next, recording strategy, just eavesdropping. You put in a different type of electrode, and this one, instead of stimulating the neurons there, this electrode tells you every time these neurons get excited all on their own. You're listening as to what it is. So you wave your hands in front of someone, and this neuron in the visual cortex suddenly gets excited with your recording strategy. Great. You're recording from some limbic structure, and you like read the most depressing part of Mice and Men or something, and suddenly neurons here start firing. We've just learned something else about what this part of the brain does. With this diagram here of an ear, you are listening, you are stimulating or your lesion. Next option in this horrendously inaccurate drawing looks nothing like. Okay, you could look at the biochemistry. What proteins are being made in neurons in this part of the brain under this circumstance? In this other part of the brain, but if and only if this is happening, which proteins are being made where. And remember, fact from our molecular lectures, that astonishing factoid there, that 70% of the genes that are only expressed in the brain, 70% of them are only expressed in certain parts of the brain at certain times. So specificity, whoa, this turns out to be the only part of the brain that makes this protein, for example, and you've just learned a ton. What is the most useful thing if you come out of the biochemical approach? Finding out what kind of neurotransmitter is being released by this neuron. What neurotransmitter is characteristic? This is a dopamine releasing neuron. This is a serotonin releasing neuron. We will get familiar with all of those. The key biochemical thing that people want to know is what's the neurotransmitter? And that was some of the fanciest stuff of the last century, first being able to figure out that there's dozens and dozens of different neurotransmitters, messengers, released one characteristic one, released by this type of neuron, and out it comes, and then what does it do? And you get lots of insights that way, and we will see plenty about that, and all sorts of drugs that influence brain function, influence behavior, influence emotions, are working on the neurotransmitter end of it. Next strategy, a molecular one. So now you look at neurons in that part of the brain and you see which genes are active under which circumstances and which genes are always silent and where there are epigenetic silencings in this part of the brain, and you don't see those silencing methylation markers in this other part of the brain. You're slowly putting a map together of, okay, destroy or stimulate or record from this part of the brain. This is what is different at that point. And this is the part of the brain, the only part or the predominant part or one of the more predominant parts that makes this protein or activates this gene or has this epigenetic thing that's gone on. You're piecing together a story there from it. Where neuroanatomists really fall in love and are so happy is to now do the next approach, which is circuitry. Who does this part of the brain talk to, send axonal projections to, releasing its neurotransmitter? Who is it talking to? And who is it hearing from? Who is it getting projections into its dendritic spines? Who's talking to it? Who is it talking to? And this is incredibly useful stuff because you begin to see the circuitry there. And this is something that you can see sort of indirectly with brain imaging techniques. When you have functional brain imaging, you can see, oh, okay, something or other happens, and this part of the brain, this part, and this part suddenly activate. And then you wait about 20 years and you get an even better functional brain scanner that could do better time resolution. And you see, whoa, this part of the brain activates, and 50 milliseconds later, this activates, and 20 milliseconds after, this activates, and you're beginning to get a sense of circuits. And by the time we get to the depression and schizophrenia lectures, we're going to see the circuitry is hugely informative. Who is driving the function of some other brain region with what downstream consequences, and where might there be something going wrong? There are approaches. The final one is to look at plasticity. How does this stuff change in response to experience? And neurons can grow new axon terminals and new dendritic branches, and neurons can change the excitability of these synapses, long-term potentiation things. Okay, so these are all the techniques you use. And you put them all together, and now instead of asking easy stuff like what do these three neurons have to do with what's going on in this hand, now you can start asking what does our PAPE circuit and all those structures and connections have to do with emotion and social behavior and emotionally driven decision making and things of that sort. So right off the bat, using all of these different approaches, you still have your hands full to try to figure out what's happening. Here's an example as follows. Okay, here's the example. Here is Stanford, and in San Jose, there is the factory that makes boba. And as we know, boba is incredibly important. And once the boba is made, it is shipped down 101 to Stanford University. And you are now trying to figure out, here sitting in Stanford, where does boba come from? What is the projection source of boba? And what you do is you go in and you do a very, very carefully neuroanatomical thing. You drop a bomb on the middle of 101 somewhere around Cupertino. And suddenly, there's no more boba arriving here at Stanford. Whoa, what have you just done? You've discovered the center of boba making. You've done no such thing. You've found the pathway. The fibers of passage is the term in the business by which the actual center is sending information down there. And if you are not careful and you jump to hasty conclusions, you publish in the literature and send research in a completely wrong direction for years by saying, aha, this is the boba center of the brain. It's not until you do more careful work, anatomical ways of visualizing these sorts of pathways, that you realize, no, this is not where the nucleus is. This is not where the cell bodies are. This is just the axonal cable. This is where it's happening. So the problem of fiber of passages, they're telling the difference between a nucleus and the way by which it's talking with the rest of the brain. More challenges that come in with that. You have to be ethologically informed. Okay, what do I mean by this? Suppose you get some part of the brain and you stimulate it and you are studying a lab rat. And what you see is you stimulate this part of the brain, and as a result of doing that, the lab rat suddenly runs around the cage like crazy where you've put a whole bunch of newspapers in there, and the rat shreds the newspapers and pushes all the little pieces over into a corner. Okay, so we've just found the recycling center of the brain or something. What is that about? Well, being a good ethologist, you decide to go study it in somebody else, and you stimulate that same part of the brain in a human. And what does the human do? The human goes and signs up to be on the waiting list for their 1-week-old child to get into a good preschool in 4 years. Or, oh, this is having to do with maternal behavior. You stimulate this part of the brain, and what you get is the fixed action pattern of maternalism in a rat, which is to shred up stuff and get little leaves and make a little nest in the corner for where you're going to have your kids and keep them safe there. You do the same thing in the brain of a rhesus monkey female, stimulate the same area, and she will grab any sort of object that's kind of oblong or something and put it up as if she is nursing. You are triggering a fixed action pattern that way. Whoa, so this is the push something to your chest, shred up the newspaper brain center. Go figure out ethologically that these serve the same purpose, but in different species. Aha, we've just found a part of the brain that has something to do with maternalism. Or another example, you stimulate a different part of the brain, and this time sitting in your lab is a lion, and you've stimulated this part of the lion's brain, and what you see is the lion extends its claws for a second. What's up with that? So now instead you get a human and you stimulate the exact same part of the brain, and what they do is very briefly they say, shit. So we're looking at the claw extending profundity brain region. What are we looking at? If you're stimulating this region very mildly, you're eliciting the first bits of aggressive behavior, of hostility, lions flexing its claws, humans giving a slightly menacing bit of language. Okay, so when a human is beginning to get irritated, they don't do this, lions do that. Know your ethological rules there in order to decode what's happening. Also, know the individuals that you are studying. For example, you stimulate one part of the brain in this primate, in this baboon, and as a result, this baboon leaps on another baboon and savages his throat. Okay, well, that's kind of dramatic. You now know that this part of the brain has something to do with aggression or something, and now you get another baboon and stimulate the exact same thing, and he doesn't leap on that guy and try to savage him. Wait, so is this the part of the brain for aggression, or is it not aggression? Who are the individuals involved? The first guy happens to be the alpha male in the group, and if he feels like attacking this guy, feel free. And the second guy is the lowest ranking guy in the group, and no matter how much he is getting aggressive urges there at that point, that's not a behavioral option. Oh, you will elicit these behaviors only in some individuals. And eventually we will get to some research done on baboons back when by my wife, showing that around the time that female baboons are menstruating, that you see something that looks like a perimenstrual syndrome of behavioral changes. And what you see is some animals get more aggressive the 2 or 3 days around their periods, and other animals get socially withdrawn. What's up? Who is it who gets more aggressive? The high-ranking females, because they have the behavioral option of being aggressive. And who are the ones who instead get socially withdrawn? And all those are the low-ranking ones who would love to aggress somebody else because they're feeling all edgy and irritable and stuff, but they don't have that option. So making sense of, ooh, in this case, what part of the brain are the hormones having to do with the menstrual period? What are they working on? Well, sometimes we see aggression, sometimes we see social withdrawal. Who's the individual you're studying it in? So remember your ethological principles and know the individuals involved when trying to make sense of what's going on in the brain. We are going to come back to these individual areas in the lectures in the second half of the course in great detail. Okay, the amygdala. The amygdala is about fear. It's about fear, anxiety, things of that sort. Back to our amygdala diagram from before. The central part of the amygdala is fears that you were prepared to learn about. Phobias, ethologically prepared learning, where it takes less conditioning to decide that you're afraid of spiders than to decide that you're afraid of textbooks or something. The outer part of the amygdala is about things you have to learn to be afraid of in a sustained way, trial and error, all of that. So the amygdala is about fear and it's about anxiety. So immediate confusion with that is something alluded to, which is when you stimulate the amygdala, the lion does this. Or when you stimulate it a whole lot, the baboon, the alpha male, leaps on the lower ranking guy and savages him. Oh, the amygdala has something to do with aggression. And here we see one of the greatest insights you could get from neuroanatomy. You could not understand the neurobiology of aggression outside the context of the neurobiology of fear. And a world in which no amygdaloid neuron need be afraid, lions and lambs would be lying down next to each other under fig trees. Oh, fear and aggression are intimately intertwined, and the intertwining of that occurs in the amygdala. So the amygdala is having that role. You see all sorts of ways in which people figure this out, and some of the most made for TV movies, HBO versions, or whatever, is you get a perfectly normal, quiet individual going about their lives, and unexpectedly, one day, they murder a loved one. And it is so from out of nowhere. Or one day, they go get a rifle and climb up a tower and start shooting at people below. And this is beyond trying to make sense of until you get the brain afterward and you see there is a tumor in the amygdala. And this gave rise to the first of the American mass murderers. This was someone who once held the record for mass murders in America, but soon passed by the younger generation of competitors, but we still salute him for his pioneering work in the early 1960s. This guy who climbed up the clock tower at the University of Texas in Austin with a rifle and opened fire on people below and killed a whole bunch of them. The first mass shooter case in America. And this was one that gave rise to a cliche with that of all the neighbors saying, oh my God, we're so surprised. He was such a nice guy. He was so quiet. He always said hello, all of that. He was like a choir boy. This guy was a choir boy. This guy actually had been a choir boy. He was a grad student in engineering at University of Texas. And for the previous year, he had been going to doctors saying, I'm having these awful headaches radiating out of this side of my head. And they all said, well, we can't see anything wrong here. Maybe you should go see a psychologist. And then for the previous 3 months, he had been going to doctors saying, not only do I have this headache radiating out from there, but I'm thinking these thoughts about doing stuff that I would never want to do. And then they all said, whoa, you really do need to go into therapy at this point. And then the morning of, he killed his wife and left a note attached to her remains there saying, I have no idea why I did this. This is the love of my life. And he went and killed his mother and left much the same note there. And he went up the Texas Tower and shot a whole bunch of people and killed them down below. And he left a note there saying, I have no idea why I'm doing this. I have no idea why I'm doing my damage, but here's the account number for my bank account. Please use my estate to help research on what the hell went on in my brain. And what was going on was, post-mortem, a tumor found in his amygdala. And there have been plenty of cases like that ever since then. So that's one example. One interesting version of that was a few years back. There was another guy who this time went up to a hotel in Las Vegas up in the upper floor and leaned out there with his automatic weapon, and there was like some country western festival going on down below, and he opened fire and produced more casualties than any other mass shooter in American history before killing himself. And his brain has been being studied over at the med school by the neuropathologists there ever since, and they have been finding something was wrong with that guy's brain, centered around some of the projections into the amygdala. So that pops up. And so that's evidence that the amygdala has something to do with it. Then there's this profoundly rare disease of forgetting its name. It's two German names hyphenated, and whatever it is, you get calcification in the two sides of the amygdala. Only the amygdala, I'm sure there's people who are figuring that out, but the amygdala gets destroyed. And this is someone who you cannot evoke fear in. You could not get a fear response out of that. And they are pathologically trusting. What does that look like? You put somebody in a prisoner's dilemma game or some such game, and they're playing against the other person, they're cooperating, and the other person starts stabbing them in the back, and your usual person says, well, jerk, I'm now going to shift my play strategy, and they respond to the negative reinforcement of the other person stabbing them in the back. Somebody without an amygdala never shifts their strategy. They're never sitting there and saying, I want to kill this person because now they are so cooperative and they stab me in the back. They don't shift their strategy. All sorts of examples of odd things like that occurring in these folks with this very, very rare neurological disorder. More evidence about the amygdala. In the 1970s, there was this major biomedical ethical storm that came up when, as people were beginning to learn about the amygdala, you go in and you surgically destroy the amygdala of a rat or a monkey and you can't evoke aggression anymore. 3 neuroscientists at Harvard Med School published an editorial in the New England Journal of Medicine, and just because that didn't infuriate enough people, they then published it in like the New York Times or something, saying, whoa, so we're just learning about this amygdala area, and you notice how there's all this urban rioting that's been going on, and all these dangerous inner city youth, and that maybe we should just go in and destroy a bunch of people's amygdalas, do amygdalectomies, and that will make for a much more peaceful society. And no surprise, all sorts of people were not pleased with that proposal. And this was a notorious incident. And whoa, thank God that didn't happen. Court-ordered amygdalectomies have been carried out on people in all sorts of settings. The largest number of cases of it was in India, but a fair number in the United States, where you get somebody who is judged as having intractable aggression and court-ordered destruction of their amygdala. So, amygdala, fear, aggression, all of that, and then we see some interesting additional things. When you stimulate the amygdala in a male rat, if you stimulate the right part at the right frequency, you don't get aggression, you get sexual arousal. You get male appetitive behavior. The male is suddenly willing to press a lever for access to a feed. male sexual arousal, male sexual motivation tied into a part of the brain have something to do with aggression and fear. And you can have a field day with that, do the same thing in the female amygdala. You don't see anything like that. So there's something going on with sexual motivation in males in the amygdala, along with fear and anxiety and aggression. Another interesting domain. So you sit down your average person in a brain scanner and you flash up various pictures of things, and you flash up the face of like something scary and menacing, and what do you know, the amygdala activates within milliseconds there. Yeah, amygdala, fear, anxiety, all that sort of thing. Now you get somebody who is clinically depressed and put them in the brain scanner, and you flash up pictures of all sorts of things, and you flash up pictures of threatening faces, and the amygdala doesn't activate. What is it that makes the amygdala activate in someone who's clinically depressed? Pictures of sad things, pictures of depressing things. Whoa, so what have we just learned about the amygdala? The amygdala also plays a role in depression. That's totally weird. Where'd that come from? And then you think about it, and depending on what species you are, ethology, redux, and depending on what sort of individual you are within that species, what the amygdala responds to is whatever it is that scares you. And if you're someone with the clinical depression, the scariest thing on earth is something that's going to make you feel even sadder. And people are now beginning to work out, how does the amygdala get roped into having this role during depression? So that tells us something about the amygdala. Next limbic system that we're going to pay a lot of attention to, the hippocampus. The hippocampus, I devoted like 30 years of my life worshiping at the feet of the hippocampus. What's the hippocampus is about? It's learning and memory. Learning, not every type of learning, not every type of memory, learning of things that you were consciously aware of, declarative knowledge, explicit memory, and what part of the brain or what's the hippocampus damaged in most meaningfully in terms of clinical neurology? Alzheimer's disease. It is ground zero for where damage occurs, and Alzheimer's disease, and a ton of work has gone into figuring out why it starts there, why those neurons are so full. Okay, so hippocampus, learning, critical for this explicit sort of learning. You don't use the hippocampus to learn how to play a piece on the piano or how to do a tennis backhand or something. That's a different part of the brain where you're consciously aware, I know this, I know that I know this. I can plan strategically based on knowing that I have that knowledge. The hippocampus is about this. So at this point, you should be stopping your tracks and say, wait, this is the limbic system. We're talking about emotion and arousal and fear and sex and love and who knows what, and memory? What's the hippocampus doing in the limbic system? Memory, and something we will begin to appreciate down the line is your memory system is not just a camera recording what's going on, it's being filtered through emotions. How intensely you remember what's happening, how accurately you remember what's happening. And this gives rise to a bit of neuroanatomy that's incredibly relevant in courts of law, which is during moments of extreme arousal and fear and rage and all of that, you file stuff away in your hippocampus, traumatic memory, flashbulb memory. Where were you when? I'll remember this for the rest of my life, but you don't remember it very accurately. And what's this about? This is during those moments of arousal when the amygdala has taken over hippocampal function for remembering traumatic stuff. And because the amygdala is not very good at like remembering new facts in time for the SATs kind of stuff, it's not great. It's not very accurate commander of hippocampal memory processes. Where is this relevant in a court of law? The fact that eyewitnesses to traumatic events have intense memories of what happened, and they will have them forever of traumatic stuff from dark, and they're very frequently inaccurate. Eyewitness testimony in courts of law is notoriously unreliable because, yeah, the hippocampus is going crazy, then it will remember it forever because the amygdala is telling it, don't bother looking at the details, just quick, remember it in a fraction of a second, and you get inaccuracies then. You cannot make sense of human memory outside of the context of we file stuff away in our memory filtered through the emotions that we feel at the time, and we retrieve the memories filtered through what we're feeling at the time as well. So, hippocampus is central to that. What else does it do? It has roles in releasing stress hormones and things of that sort. Hippocampus, what it's mostly about is this role in filing away memories. And this role for the hippocampus was first appreciated in humans in the 1950s with the most famous neurological patient of all times. Okay, any of you who have had sort of intro neuro stuff will know actually the most famous patient was Phineas Gage. And we might hear the tales of Phineas Gage down the line here, but this was the second most famous neurological patient of all time, somebody only known in the literature by his initials, HM. HM is the only human in history where people went in and intentionally surgically removed his hippocampus. Whoa, what was that about? HM had a type of intractable epilepsy as a teenager. This was horrendous. You normally have an epilepsy, a single spot in the brain where seizure initiates, an epileptic focus. In this case, he had two epileptic foci, one in each of his hippocampi, and one would have a seizure, which would trigger a seizure, which would trigger, and he was just having clusters of seizures, something called status epilepticus, clusters of seizures that were going to kill him at some point, and none of the good anti-seizure drugs that are used now were available yet, and people were able to figure out this was happening. It was in his hippocampus, and they did this Hail Mary thing of saying, okay, let's go in and surgically remove his hippocampi and save his life that way. And people knew beforehand from 30 years of rat studies and monkey studies and all of that, the hippocampus has something to do with forming memories. And they said, well, he's going to have problems with that afterward, but we need to do this to save his life. And he consented to it, and his parents, who were still his legal guardians at the time, consented to it. And a famous neurosurgeon in Wilder Penfield went in and took out his hippocampi, and it mostly solved HM seizure problems. It definitely saved his life in that regard. And it wasn't until soon afterward that new drugs were introduced that would have prevented him from having to go through the surgery. But in any case, what happened to HM when they took out his hippocampus? For the rest of his life, he never formed a new explicit memory. For the rest of his life, he had a 30-second memory span. He remembered everything from beforehand because that information, when learned via the hippocampus, was then stored up in the cortex, that kind of thing. But he was never able to form a permanent memory ever again. And as described by one of the scientists who spent decades working with him, he was perpetually in present tense. There was never anything filed away, and he had a 30-second memory span. And I got to meet HM once, and what I'm supposed to say now is, of course he won't remember me, but HM, I met him once. I was working in a lab at MIT. I was still an undergrad and sort of sitting there, and HM lived in a nursing home in Connecticut and basically belonged to a bunch of neuroscientists at MIT. And every few months or so, they'd bring him up from his place in Connecticut and do testing on him. So today was the day that HM was coming in for testing, and I'm sitting there like somebody in the lab runs in and says, guess who's out in the hall? Guess who's out in the hall? HM. HM, oh my God, let's go meet HM. This is like taking Pavlov's dogs for a walk in the park or something. Let's go meet HM. And then we run out in the hallway, and there's this like middle-aged guy and the researcher who is sort of like watching over him that day. And we go up, and he's this like totally friendly guy. He says, "Hi, I don't believe we've met. My name's Henry." And we shake his hands, and we like, I don't know, if you begin to talk about the weather or something, and somebody like slams a door down the hall. There's something where we all glance over for a second and then look back, and he said, "Hi, I don't believe we've met. My name is Henry." 30 seconds and it was gone. And you could have stood there all day long with him introducing himself to you every 30 seconds, because none of it was filed away. And all he knew is, oh, here's the new face of a person. He had somebody who helped take care of him for years and years in sort of his residential facility, where he probably introduced himself to that person 600,000 times over the course of the decades there, because he didn't file anything away. Think about what life was like for him. Think about emotionally, every single day, inevitably, he would ask someone, "When is my mother coming to visit?" And umpteen times a day, he would be told, "Actually, Henry, your mother died some years ago." That as a repeated experience every day. Every morning, he would go into the bathroom and look in the mirror, and he looks like a 50-year-old guy because he was 50-year-old, and all he knows is that he's still like a late adolescent, and he like, what happened to my face? And what you see is, what's frequently the case with neurological patients like these, is you see them grasping for an explanation for why the world has done this incredibly odd thing to them. I saw a film once of HM going into the bathroom in the morning, and this was one of the people who stayed with him, helped him out filming him. And he goes into the bathroom to like wash his hands, and he's washing his hands, and he glances up at the mirror, and he glances up again, and he's like 30 years older than he remembers himself as seeing, and he looks in the mirror for a while, and the person says, what's up, Henry? What's it? And he says, "Oh, I don't know. I must not have slept very well last night. I look terrible. I look like a 50-year-old guy when I'm only 18, looking for attribution. Whoa, I must have slept very poorly last night." A hugely emotionally disruptive world to him. And what was most remarkable was when he was being told 10 times a day that, "No, actually, Henry, your mother died some years ago," he would say, "Oh." He would not react emotionally. And a whole debate was, did the hippocampus have something to do with those sorts of emotions? Or is this some sort of protective thing that he had to do over his decades like this of getting emotionally shocking news every day, of just blunting his response to everything? And at one point, I almost got permission to look at stress hormone levels in him, but he had a seizure around then, and they decided they needed to change his meds, more of his residual seizures, and all sorts of stuff was filed at that point. But this incredibly bizarre life of this was someone who could not learn anything new, until it turned out that he did learn some new stuff. He learned some new things in very interesting ways. He learned how to do things. He learned motoric stuff. He learned how to take a puzzle apart and put it back together and do it faster over time. He learned things that were not declarative and explicit. He learned things that were procedural and implicit. And this gave rise to this big shift in the field of recognizing there is explicit and implicit memory. Explicit, you learn facts and you know that you know the facts, and you know that you learn. Implicit, stuff that your body has learned better than your mind has, motoric stuff. You're using implicit memory when you get good at a sport. You're using it when you're playing a musical instrument without having to think about what the next note is you're going to play. Implicit, and it turns out that's in a different part of the brain. And implicit learning was preserved in Henry, so that he would sit there and you would give him like this puzzle thing to take apart and put back together. And it's like difficult. You need to do it a whole bunch of times. And he's saying, I don't know, I've never seen this thing before. I have no idea at all. And he's taking it apart and putting it back together because he's done 50 times before. He's learned to do it just as fast as somebody who could say, yeah, I know this one. This is the 50th time I'm doing it. He had no explicit awareness he had ever seen this thing before, yet he learned implicit stuff. Another example of him trying to make sense of the world around him. And this was a friend of mine who is one of the people studying HM. And HM was up for sort of some of his testing, and MIT at the time had sort of a clinical center there that HM would stay in there for his few days of testing. And one day, my friend had been finished up some testing with HM and was walking HM back to the place he was staying in. And he said, suddenly he got in his head saying, Henry, I know you don't remember this, but you actually stayed here last night. Do me a favor, when we get out of the elevator, just walk down the hall and tell me if you remember which room is yours. And they get out, and Henry's walking, and my friend is staying behind, so he's not inadvertently cueing him. And Henry's walking down, and he suddenly stops and he says, That was my room. My friend says, oh my God, Henry, you're right. How do you remember that? And he said, that's my name on the door. So he busts out laughing at that point, and my friend's laughing as well. And okay, pull off a fast one on the egghead in the lab coat or whatever, and like they're laughing about it. And my friend sort of waited about 15 seconds or so, and Henry was still laughing. And my friend said to him, hi, what are you laughing at? And Henry reached for some sort of explanation because he had already forgotten this, and he came up with some totally lame knock-knock joke or something. The emotional state of the humor was carried on, yet the attribution, the explicit memory of what triggered it was gone, and he just had to search for some sort of explanation. HM died a decade ago, 2 decades ago, and it was essential that his brain be studied, if nothing else, to confirm that the surgery was done in the right place, because if the surgeon missed and actually took out his nose instead, and for 40 years people have been thinking the hippocampus did something and it was actually the nose doing it, they needed to confirm the lesion was in the right place. And this produced the closest thing I've ever seen to like a sacred ritual in neuroscience. His brain was sliced by some neuroanatomists at UCSD, and they sliced it over the course of days and days, taking thousands and thousands of thin slices there and preserving them, all of that, and they live streamed it. And for those days, if you were a member of the Society for Neuroscience, you could put in your ID number or whatever, and you could watch HM's brain being sliced there. HM's brain on a block of ice being sliced, and people just sat and watched this and came back to it the next day to see what they were up to. And somehow watching it, you could be watching it with 50 other people and you would all be silent. Again, this was like the closest thing to like a religious ritual or something for neuroscience, the slicing of HM's brain. So hippocampus does that. What some other brain regions? Septum, insofar as the amygdala is about aggression, the septum is about inhibiting aggression. And suddenly that circuitry, no wonder when the amygdala is getting all excited and taking over the hippocampus and aggressive, fearful memories, no wonder it's got that striae terminalis thing looping around straight into the septum. It's trying to inhibit the septum. They work in opposition. Amygdala about aggression, septum about the opposite, and big surprise, go in and surgically destroy somebody's septum, and you get uncontrolled rage, what was called septal rage. You have a lab rat with a septal lesion, you open the cage and the rat like leaps for your throat and all of that, and because the brakes on the system had been cut. Some other areas that are relevant. Prefrontal cortex, that is so relevant and so interesting that all of Friday's lecture is going to be about that and about its interactions with the limbic system. Then those two areas, the ventral tegmentum and the nucleus accumbens, hang on, let's switch back for a second here, and there it is, the ventral tegmentum and the nucleus accumbens. We flip back this way, and what are they doing? And it's clear what they were doing. Everybody's known for decades what they were doing, which is they are about reward and pleasure. The ventral tegmentum is the heart of where your brain releases the neurotransmitter dopamine. There's other parts of the brain that use dopamine, but the ventral tegmentum is dopamine, neurotransmitter. Everybody knows about dopamine. Dopamine is about pleasure. It's about reward. Cocaine works on dopamine synapses. All sorts of other euphoriants work on the dopamine system. Dopamine is about reward and pleasure. And thus, the ventral tegmentum talking to the rest of the limbic system by way of that mesolimbic dopamine projection, and talking to your frontal cortex by way of that mesocortical dopamine projection. Your ventral tegmentum is all about reward and pleasure. And that seemed to absolutely be the case. Take a person or a monkey or a rodent or whatever, and they're sitting there, and from out of nowhere, you suddenly get a reward. There's a burst of dopamine released from the ventral tegmentum. It's about reward, straightforward. And then people figured out that's not what the ventral tegmentum, that's not what the dopamine system is about. It's not about reward, it's about something much more interesting than that. Okay, so now you set things up where instead of just giving this individual an unexpected reward from out of nowhere, you set him up, this person or monkey or rat, where a little light comes on in the room. And that's a signal telling you now, if you press a lever 10 times, you will get a food pellet, you will get an M&M, you will get a piece of kibble, whatever. A signal saying we're starting one of these work sessions. You do the work, you get the reward. And fairly quickly, the subject learns about this relationship. So when does dopamine suddenly surge? What this suggests is as soon as you get the reward. That's not when you get the dopamine. Where you get it is instead the second the signal comes on. What is that about? That's about sitting there saying, yeah, I know what the signal means. That means if I press the lever, I'm going to get a reward. Yeah, I know how to do this. I'm like a lever master. This is going to be great. Whoa, I'm on top of this. This is going to be terrific. Dopamine in this setting is about reward, but in the much more common setting, it's about anticipation, about this is going to be great. This is going to be fantastic. Dopamine is about anticipation. And critically, if you block this rise of dopamine and it just stays flat there, you don't get the lever pressing. It's not just about anticipation. This is going to be fabulous. It's about feeling motivated enough to do the goal-directed behavior then to get the reward. So sometimes it's about reward, but much more fundamentally, it's about the anticipation of the reward, the work you're willing to do to get it. It is not about the pursuit of happiness. It's about the happiness of pursuit. And that's really, really informative. Most of the time, what's going on here is this is the neurobiology of this is going to be great. This is going to be fantastic. And this reminds me of one of the most profoundly cynical things I've ever heard anyone say. This was a guy down the hall in my dorm in college who once said, a relationship is the price you pay for the anticipation of it. Whoa, is that screwed up or what? But this was already decades in advance understanding. Dopamine was not about the relationship. Dopamine was about how great it was going to be as soon as you knew it was going to happen. Yes, yes, he had the most screwed up social life you can imagine. But dopamine in the ventral tegmentum about anticipation and the work you were willing to do. And what we're going to see on Friday is the frontal cortex is really good at making you do things that take self-discipline. It's the ventral tegmentum projecting into the frontal cortex. The ventral tegmentum is saying with dopamine, this is going to be great. This is going to be great. Do the work. Do the work. You're going to enjoy it. It's going to be worth it. All of that. That's where you're getting that drive from. Okay, an important elaboration here that explains an awful lot about what happens in lots of our lives. So in this scenario, here we've got the signal comes on, you do the work, you get the reward, guaranteed 100% of the time. Now, let's change things so that the signal comes on, you do the work, and only half the time you get the reward. You can't predict it, but it averages out to only about 50% of the time you get the reward. What happens to dopamine at that point? As soon as the signal comes on, dopamine release looks like this. It goes through the roof. It's totally gigantic. What is this about? We've just inserted a very critical word into our neurochemistry. This is the dopaminergic neurochemistry of the word maybe. Maybe. Whoa, does that drive us? The signal comes on and you're feeling great because, whoa, I know how the signal lever pressing things work. I'm on top of this, but I'm a total screw up. But today I'm wearing my lucky underwear, but oh, somehow things always go. And you're just teetering on this fulcrum of 50% chance. And that boosts out dopamine like crazy at that point. And what's that about? That's what the neurobiologists who designed Las Vegas understand. Unpredictable intermittent reinforcement gets work, gets anticipation, gets motivation out of us like nothing you can imagine. And here's an elegant demonstration of how that really is the case. Now we have either 75% likelihood of reward or 25% chance. What happens to dopamine? It does that. That's crazy. Wait, 75%, that's great news. You're getting closer to guaranteed reward. 25%, that's terrible news. You're that closer to never get. How can they get the exact same curve? Because they have the exact same degree of unpredictability. 100%, no uncertainty. 50%, the maximal uncertainty that's possible. 75 or 25, about halfway in between. So this is spectacular, and what you should realize is when you think about what they do at Las Vegas is they have ways of psychologically manipulating you so that you feel like you have like a 50% chance, and this is good, when in reality you've got 1,000th of a 1% chance. But they've psychologically manipulated you into deciding today is your lucky day. Today is the day that we've had all sorts of ways of making. We've given you free hors d'oeuvres, and we said because you got a lucky number on your register, we've given you free drinks to make your judgment even worse. You do something that is done in Las Vegas hotels, which is all sorts of floors are not numbered. That was stated poorly. You get into the hotel there, and there's the first floor, which is called the first floor, and the second floor, and the third floor, and the fourth floor they call the 15th floor. And the fifth floor, they call the 16th floor. And what does the person at the desk say? Oh, congratulations, your registration thing here was a lucky number. We're putting you in one of the upper suites on the 16th floor. And you go into the elevator, and it doesn't tell you that you're actually going to the fifth floor. You get out, and who looks out the window because you're already drunk and you're not being able to make much sense of depth perception. It's, whoa, my lucky day. I just got this free upgrade to the upper, and all they're doing is manipulating people to distort where this actually goes, and thus to get more and more work out of you. Okay, a little bit more on dopamine and reward, and we'll be returning to all of this. Here we see another feature about dopamine and anticipation, and this accounts for like such a percentage of Earth's misery and stuff. So you got somebody and you did something special with them yesterday. They're sitting there and unexpectedly you give them a reward from out of nowhere. And what happens? You know what happens already? They've got this surge of dopamine. And that's great. Whoa, how'd I get like 2 pellets instead of one, twice the reward of what I was expecting? That's fantastic. Now you come back to them today and you give them the same reward, and this is what dopamine does. And tomorrow you give them the same reward, and this is what dopamine does. What's that about? What was wonderful and unexpected yesterday is what you feel entitled to today and isn't going to be enough tomorrow. And this habituation is why the more we consume, the hungrier we get. Why we just want and want and want, because this system habituates incredibly quickly. Okay, few more brain regions. Hypothalamus, in our closing minutes here. Hypothalamus has all these different sub-areas in it. 2 areas we're going to hear a ton about. One called the ventral medial hypothalamus, the VMH. Don't worry, you will become familiar with these acronyms. The other called the medial preoptic area of the hypothalamus. What are they about? Sexual behavior. What are they about? Sex differences in sexual behavior. What are they about? All sorts of interesting differences that may or may not correlate with sexual orientation, with sexual identity, things of that sort. So we know lots about those areas. Another area we're going to hear next to nothing about ever after, something called the suprachiasmatic nucleus. And if you say it loud enough, you'll sound precocious. What's it about? It's about circadian rhythms. There's another part, the paraventricular nucleus, that's about initiating the stress response, initiating the hormonal stress response. So it's sitting there at the base of the hypothalamus, talking to your pituitary. Then another area called the lateral hypothalamus. And this is one of those ethology coming back with a vengeance, because people lost about 20 years of work on the lateral hypothalamus as follows. You take a lab rat and you stimulate the lateral hypothalamus, and what does it do? It jumps on a mouse that you've put in the cage and savages it and kills it and rips it to shreds. And whoa, okay, lateral hypothalamus. That's all about aggression. And now you take a human volunteer and you stimulate the lateral hypothalamus, and they leap up and they grab a box of Cheerios and shred it to pieces in order to eat the whole thing. Wait, that's not aggression. That's not going on there. It's about acquiring food. If you're a rat, you eat mice. That's about predation. If you're human, you do that, and you just open up a box of cereal instead. And what we got was people being steered way off course for years. Back when I was in diapers and learning about the hypothalamus, they would teach you the lateral hypothalamus is the center for aggression. No, not at all. And eventually they figured that one out. It is the center for keeping track of your blood glucose levels and your triglyceride levels and how long ago you ate a meal and whether or not you're smelling stuff that smells appetizing and all. So ethology taught people a lesson there. If they had known that, they would not have wasted about 20 years of research on that mistake. Okay. Any other parts of this? The last part here, this critical thing is, in addition to this picture of top-down regulation and bottom-up regulation, all of that going into your brain deciding what it's going to tell your body is also telling your brain what emotions to feel. At least that was the theory back when, about 120 years ago, the philosopher William James of the last week's ethology, history stuff, William James came up with something called the James Lange theory of emotion. I don't know who Lange was, but he got lost in the dust of history's dustbiners. William James, the James Lange theory, as follows. What William James said is, when you feel an emotion, you really have not just felt it in your brain. What happens is something emotionally meaningful happens to you, around you. You see something scary, something happy, something arousing, whatever. And as a result, your brain affects the workings of your body. And then you sit there, your brain senses these changes in the body and says, oh, if these changes have just happened, I must be feeling this. That the bodily responses to emotion come before the emotion themselves and in fact instruct what emotions you have. Okay, so people pretty quickly laughed this out of the room because, for one thing, the bodily changes take seconds to occur and you feel the emotions immediately. And the other is whether it's fear or arousal, and we'll get to this in the aggression lectures, what's going on in your body there. You can't distinguish between somebody who is in a murderous rage and someone who has had an orgasm. You don't get the specificity. Okay, so James Lange, they were wrong. Your brain is not deciding what emotion you're feeling based on this feedback. So that got tossed. But what people have since learned is, your limbic system comes up all on its own with what emotion you're feeling. What this feedback, what came to be called interoceptive sensations. How fast is my heart beating? Am I short of breath? Any of this stuff. What interoceptive information is influencing how intensely you feel the emotion. And this is our whole world from last week, where you give somebody adrenaline and they don't know it, and as a result their heart is beating faster and they're breathing faster, and they assess the attractiveness of someone who's attractive as being even more attractive. It is a William James sort of moment there where the limbic system is deciding all on its own how attractive this person is or isn't attractive, but it's saying, whoa, but my heart is beating incredibly fast, so I must be feeling this more intensely. And you see this in a number of other realms. For example, people with clinical depression, one of the all-time easy interventions, which isn't curative but helps a little bit, is you very mechanically, artificially force the person to smile. And smile again, and there is some sort of interoceptive feedback loop from these muscles back into the brain. And in some way, the brain is saying, well, yeah, the world still sucks and I'm still depressed, but maybe if I'm doing all this smiling, maybe it's not quite as bad. And that could lessen the severity of depression for a little bit. Final example. So you have this very puzzling thing. You've got somebody who's got a terrible anxiety disorder, and a psychiatrist prescribes for them something called a benzodiazepine, an anti-anxiety drug, Valium and Librium, and it lowers anxiety levels. Meanwhile, across town, you've got a champion Stanford gymnastics somebody or other who is pulled a muscle and they're getting muscle spasms, and they go to the team doc, and the doc gives them benzodiazepines, the exact same thing to relax their muscles. Okay, wait, so is this an anti-anxiety drug, or is this a muscle relaxant, or what's the deal? And people eventually figured out this was a James Lange sort of moment. Part of what your brain is deciding when deciding if you're anxious, how anxious you're feeling, is saying, well, how uptight is my body? How tense are my muscles? And what you see is you got the person sitting there and you give them massive amounts of benzodiazepines, and they're sitting there and explicitly, consciously, cognitively, they know the world sucks just as much as it did half an hour ago, but their muscles are so relaxed they're practically dribbling out of their chair. And what does the limbic system decide? Oh, if my muscles are this relaxed, I must not be feeling that anxious about things. the feedback loops into all of this. So not only top down in the brain and bottom up in the brain, but this whole feedback loop by which your body is telling your brain how intensely to feel emotions. Okay, so what we are ready to pick up on now, the point of this last part being why everybody wants to control what's coming down here in the looping back. What Friday will be about is why everybody in the limbic system wants to tell the frontal cortex what to do and what the frontal cortex is doing back to the limbic system in turn.